Hello and welcome to the Precision BioLogic CRYOcheck Lupus Anticoagulant Products and Introduction to HexLA webinar. So, my name is Jim DeMace. I'm the Senior National Technical Sales Manager here at Precision, and I'm joined today by Ian Burns, our Product Manager, and together we are very excited to present this information to you today. These are interesting times that we're all living in right now. Many of us working from home, working remotely. We thought it would be a nice touch just to tell you all where we're coming from today. Ian is based in Dartmouth, Nova Scotia, coming from his home, and I'm based in Santa Fe, New Mexico, coming from my home. So I think we have all five time zones represented here, Ian, today, and so we're excited to have such a wide variety of people joining us. So before we get started into the presentation, we would just like to remind you how you can ask us a question. So as we come to the end of the webinar, we'll be able to take your questions. You can simply go to the GoToWebinar control panel. It'll be on the right side of your screen. You should see something on the right side of your screen now. And there's a section there for asking questions. And we want to answer as many questions as we can. If we don't get to them, we'll make sure that we answer your questions via email. And for you also, if you think of a question after the presentation has ended, Ian and I will have our email addresses at the last slide for your convenience. So with that, let's get into what we're going to do today with you and give you a little bit of an idea of how the presentation will flow. So the first part of the presentation will be an overview. What is the antiphospholipid syndrome? And I will conduct that first part of the presentation. Ian is going to bring us through lupus anticoagulant history and testing, as well as the original history of the hexagonal phase phospholipid assay. Then I'll come back in and talk about our complete suite of lupus anticoagulant products and introduce you to our new CRYOcheck HexLA. So with that, let's get started. So what is antiphospholipid syndrome? It's abbreviated APS. APS is an acquired autoimmune disorder characterized by the presence of antibodies directed at phospholipids, a type of fat found in all living cells and cell membranes. When antibodies attack phospholipids, this causes cell damage, which can lead to the formation of blood clots in arteries and veins. Now, APS is associated with an increased risk for venous as well as atrial thrombosis, stroke and heart attack, as well as recurrent pregnancy failure. Examples like late-term miscarriage, premature birth. Now, sometimes it's associated with a specific autoimmune disease. A most common example would be the systemic lupus erythematosus, but then there are times when it's presented in isolation as well. So what causes antiphospholipid syndrome? It's not known what causes the immune system to make antibodies against phospholipids. Now, it is well documented that the presence of antiphospholipid antibodies can cause clots to form, yet that mechanism of thrombosis is not clearly defined. Now, there are some theories, though, perhaps the antibody action against specific proteins, the antiphosphatidylcholine and antiphosphatidylserine, perhaps platelet activation that could be due to surface disruption. You could have activation of vascular endothelium, and you also could possibly have the complement activation. There's been a recent focus placed on anti-beta-2 glycoprotein 1 antibodies. They are a subset of the anti-cardiolipin antibodies, and that's due to a strong association with thrombosis. Now, just talk a little bit about the types of the antiphospholipid antibodies. They can be classified in several different groups. I just mentioned the anti-beta-2 glycoprotein 1. which is a subset of the anticardiolipin antibodies. You could have antiprothrombin, antiphosphotylserine antibodies, as well as lupus anticoagulants. And the most common antiphospholipid antibodies are the antiphospholipid antibodies, excuse me, as well as the lupus anticoagulants. Now, because of the mechanism of thrombosis, it can vary by the antibody type. So testing for antiphospholipid syndrome presents many challenges for you in the clinical laboratory, and it must be managed through a combination of solid phase antibody assays. Example would be an ELISA type assay, as well as clot-based coagulation assays. Lupus anticoagulant is tested for by a minimum of 2 coagulation tests that are phospholipid dependent. And due to the heterogeneous nature of lupus anticoagulants, no single laboratory test can be used to reliably detect lupus anticoagulants. And Ian's going to go more into the CLSI guidelines and talk to you more about the testing protocols. Now, what is lupus anticoagulant? We in COAG, this special COAG labs, we talk a lot about lupus anticoagulants. In the late 1940s, an antibody was discovered in some lupus erythematosus patients that prolonged the clot time of phospholipid-dependent clotting tests. At that time, it was concluded that the presence of this antibody would lead to an increased tendency to bleed. But you see, lupus anticoagulant is actually a misnomer. It's often found in patients, about 50% of the time, with conditions such as systemic lupus erythematosus, but it's associated though with thrombosis in vivo, not bleeding. So hence that misnomer, lupus anticoagulant. Lupus antibodies are heterogeneous and they may also be transient. So that's the main components there. What I'd like to do now is turn this over to Ian so he can review the lupus testing and some of the history behind this and the hexagonal phospholipid assay.
Thank you, Jim, and thanks for that great overview of antiphospholipid syndrome and lupus anticoagulant. As Jim mentioned, I'm going to now dive into some of the background on the history for testing for lupus anticoagulant. Today I'm going to stick mostly to the CLSI guidelines. Now there's a number of bodies that do publish guidelines for the testing for lupus anticoagulant. ISCH also publishes a widely followed guideline around this, but just for today's session I will stick to mostly the CLSI guidelines. I will mention a couple places where the guidelines do agree on recommendations. So very broadly speaking, the CLSI guidelines break down diagnosis and testing for lupus anticoagulant into these 3 sections. Now, I won't get too technical here. I'll try to keep this fairly high level. So the first thing that's recommended is a screening assay, and it's recommended by the CLSI that you use 2 separate screening tests that represent 2 separate principles, such as targeting 2 separate coagulation pathways. If you receive a positive result in your screening assay by either method, it's then recommended that you move on to a confirmatory assay. Now you'll see as we go through the next few slides, the common thread in a confirmatory assay is the addition of excess phospholipids that interact and have a neutralizing effect on any lupus anticoagulant present in the sample and will shorten the clot time of the abnormal screening assay. So the abnormal screening assay may be your APTT assay, and the confirmatory assay would be an APTT with an excess phospholipids, and you would look for a shortening of that clot time upon the addition of phospholipids to the sample. So some confirmatory assays might be paired, DRVBT, the dilute Russell's viper venom test, or silica clotting time would be examples of paired assays. There are also independent confirmatory assays, so PMP, platelet neutralization procedure, would be an example. And there's also a category of tests that are referred to as integrated assays. An example of that would be the hexagonal phase phospholipid neutralization test. Now, if you run your screening and confirmatory assays and you're unable to show the phospholipid dependence of the antibodies. So you apply phospholipids to the sample but do not see that reduction in clot time, you may move on to a mixing test to try to further guide diagnosis. So a mixing test is the addition of pooled normal plasma in a 50-50 mix with the patient's sample. And the idea here is that the normal pooled plasma will either correct for a factor deficiency or it will be adversely affected by an inhibitor that is present in the patient's plasma, further guiding diagnosis. So the mixing test can be applied to the screening or confirmatory portion of your assay. It should be noted that if you're running an integrated assay, such as hexagonal phase phospholipid neutralization test, the mixing step is an integral part of that assay, and there's no need for a separate assay. And I'll talk about that a bit in the coming slides. So let's just go through the different categories as defined by CLSI of screening and confirmatory tests for lupus anticoagulant. So first we have the independent screening test. That would be an assay with a low level of phospholipids that shows a prolonged clot time. So the most common test in this group would be simply an APTT with an LA responsive APTT reagent. The next category of test is independent confirmatory test. So that's an assay with an increased level of phospholipids, typically still APTT based. And the idea is that in this stage of the assay, you would show a shortening of that initial clot time upon the addition of phospholipids. There's also paired assays where the screening and confirm assays are the same, just repeated in the second step with an excess of phospholipids, but using the same reagents. And then finally, we have the integrated assay. As I talked about, that would be the hexagonal phase phospholipid neutralization test. And the integrated assay contains the screening, confirmatory, and mixing tests all performed in parallel as part of the same test system. So let's look at each of these categories in a little bit more detail with an example of an assay that would be in one of those categories. So first we have the APTT, or activated partial thromboplastin test. It's based on the activation of the intrinsic pathway. It's important that the APTT reagent used here demonstrates a responsiveness to lupus anticoagulant. It's recommended as a first-line test by both the CLSI and ISTH, and it could be paired with platelet neutralization procedure as the confirmatory portion of the assay. So this is a widely used, very common test, and it's the initial screen where you would detect an abnormality in the sample by way of a prolonged clot time. Next, we have an independent confirmatory test. As I mentioned, an example here would be the platelet neutralization procedure. So very similar to an APTT test. Well, it is essentially an APTT test, but you run it in two tubes, one with a buffered saline solution, and in the second tube, you have freeze-thawed platelets as your source of phospholipids. And again, you're looking for that delta, and the results are expressed in seconds, the difference between tube 1 and tube 2, and that's compared to the laboratory established cutoff. And you're looking for the shortening of that clot time upon the addition of phospholipids or the freeze-thawed platelets. So let's talk about paired screening confirmatory tests. So the most commonly known test in this category would be the DRVVT test, or the dilute Russell's viper venom time. It's based on the activation of the common pathways. So Russell's viper venom directly activates factor 10, and it bypasses many of the other factors that might interfere with the test by having abnormalities in one of the other coagulation pathways. So because of that, it is considered more specific for lupus anticoagulant than some of the other paired assays, such as Kalin clotting time or dilute prothrombin time. And the results of this assay are expressed as the ratio between tube 1 and tube 2. So if you see in the diagram here, you have the same Russell's viper venom reagent in both tubes. Tube 1 has low levels of phospholipids, where tube 2 has the excess of phospholipids. And you're looking for, again, the shortening of clot time between tube 1 and tube 2, indicating the neutralizing of any lupus anticoagulant that might be present in that sample. Then we have the integrated screen and confirm test. So the example here is the hexagonal phase phospholipid neutralization test. Now, as I mentioned, it does incorporate the screening, confirmatory, and mixing step into one assay system. If you look at the diagram here, it does show the addition of a, I skipped a few slides there, excuse me. It does show the addition of a normal plasma in both tubes, which would correct for any factor deficiencies that might be present in the sample, helping further guide diagnosis. And the results of this assay are expressed as the delta in seconds between tube 1 and tube 2, and that would be compared against the laboratory established cutoff. And lastly, we have the mixing test. This is pretty straightforward. It's simply a 50-50 mix of pooled normal plasma and patient sample. It could be used on the screening or confirmatory steps of the assay. And the idea here is that if you were unable to show phospholipid dependence in the confirmed stage of the assay, you would run a mixing test, and that would help guide diagnosis by either correcting for a factor deficiency or perhaps indicating the presence of another inhibitor. The CLSI publishes some great diagrams that show the testing algorithms for the different categories of lupus anticoagulant assays. So I just want to walk through those quickly. This shows the algorithm for an independent system. So if you see here, we have the screen assay, which could be the APTT or the Kalin clotting time. And you're looking for a prolonged clot time if lupus anticoagulant are present. If you don't see that, you would probably stop there and determine that lupus anticoagulant has not been detected. If you do see that prolonged clot time, you would then move on to a confirmatory assay. So the example given here is platelet neutralization procedure. And then you're looking after that is the delta between tube 1 and tube 2 abnormal. If it is, you would determine that LA is present. Now the guidelines do recommend that you repeat testing in 12 weeks before making a diagnosis of lupus anticoagulant. Now if that delta on the confirm assay is not abnormal, you would likely then go back and try to explain that prolonged PTT test by running a mixing test on the screen. And you're looking for a correction. If it does correct, you would assume that LA is not detected and proceed to a specific factor assay. Now, if there's no correction, it could be a number of things. You would investigate for possible strong LA, lupus anticoagulant with a factor deficiency, or possibly another inhibitor. For the paired system, it's similar. So we have the screen, and if it's prolonged, you proceed to the confirm portion of that assay, and you're looking for a correction. If it does correct, you're showing that phospholipid dependence of the lupus anticoagulant. So you would conclude, yes, LA is present. And again, repeat testing in 12 weeks. But if there's not correction on the confirm portion of a paired system, you would then run a mixing test on the screen. If that corrects, you would say LA not detected and likely proceed to a specific factor assay. Then you could perform the mixing test on the confirm as well. And this would tell you a couple different areas that you could potentially investigate. So in this scenario, we're saying that there's no correction after the mixing test on the screen. And then if there is a correction on the mixing test of the confirm, that could be possibly LA with a factor deficiency, or it could be strong LA or potentially another inhibitor if the mixing test on the confirm does not correct. And then lastly, we have the integrated system. So I think you'll probably notice right off the bat that it's a much simpler algorithm because everything is integrated into one test system. There's a lot less gates or points where you need to move and go to a mixing study, for example. So with the integrated system, you would run the sample in 2 tubes in parallel, and you're simply looking for that positive or negative result, whether the delta between tube 1 and tube 2 is above or below the laboratory established cutoff. If it is, you would say, yes, LA present. We demonstrated the phospholipid dependence of the lupus anticoagulants. If it does not, you would say LA has not been detected, and you would likely move on to a factor assay because you would, at this stage, likely have already run an APTT with an abnormal result. So you would have to investigate the cause of that abnormal result. So again, repeat testing in 12 weeks before making a diagnosis of lupus anticoagulant. Now I just want to talk a little bit more specifically about hexagonal phase lupus anticoagulant testing. Some of the foundational work on this was done in 1986 by Dr. Rausch and her team, and they demonstrated the ability of hexagonal phase phospholipids to neutralize the anticoagulant activity of 11 lupus anticoagulant antibodies from patients with systemic lupus erythematosus. In contrast, they ran these same 11 LA antibodies using lamellar phase phospholipids, and those phospholipid structures had no effect on the anticoagulant activity of those antibodies. So Dr. Rausch and her team concluded that antibodies are able to distinguish between different phospholipid structures, and the hexagonal phase phospholipids showed a superior immunogenic response. And here is the table from that 1986 study, just showing the 11 different antibodies along with the control. And you can see clot time plotted on the Y here. And the two white bars represent these antibodies as tested by two different sources of lamellar phase phospholipids, where the darker bars represent these antibodies tested by three sources of hexagonal phase phospholipids. And as you can see, across all 11 antibodies, the hexagonal phase phospholipids were able to demonstrate that phospholipid dependence, where the lamellar phase phospholipids were not. So what does this all mean, and why would someone want to use a hexagonal phase phospholipid neutralization test? Well, I think it comes down to several things, and I've laid them out here. So first, there's guidelines. Using an integrated test such as hexagonal phase phospholipid neutralization test does satisfy many of the guidelines laid out by both CLSI and ISTH in a single assay system. So it has the screening, confirmatory, and mixing tests all performed in parallel as part of the same system. Sensitivity is often discussed with hexagonal phase lupus anticoagulant tests. It's been noted that it's able to pick up weak LA. So the case that's often given, or the example that's often given is a patient has a clot line that is above their baseline, but does not exceed the laboratory's reference interval. It may not get picked up by an APTT-based screening assay alone, but where hexagonal phase testing does not look necessarily at the clot time, but only the delta between the two tubes, and the two tubes are always run in parallel at the same time, it may be able to pick up the presence of lupus anticoagulant where an APTT-based screening test alone would not. There's specificity. So as Dr. Rausch and her team showed in their 1986 paper, and also Dr. Triplett demonstrated in 1993, hexagonal phase phospholipids are more specific for lupus anticoagulant than lamellar phase phospholipid arrangements, which does improve the specificity of the assay. And there's simplicity. So as we saw in that testing algorithm, there's really not a lot of decision points in the algorithm. It's a qualitative test that will give you a positive, negative result, and then there's a lot less ambiguity around the interpretation of results and how to proceed given either a positive or negative result at either stage of the assay system. So what's commercially available for hexagonal phase tests? There's currently two tests that are commercially available. One is from Stago, and they sell that under the brand name Stakelot LA. This has been available for a number of years, and it's available in two formats as a lyophilized reagent set. That's Stakelot LA10 and Stakelot LA20. Same vial count in both of those kits, just different aliquot volumes in the vials. And also, we're happy to announce, and Jim will certainly dive into this in a little bit more detail, we launched in February of 2020 our CRYOcheck HexLA kit. It's licensed by Health Canada, it's CE marked, and it's also on the Australian New Zealand Registry of Therapeutic Goods. So it's available in all of those markets. We do currently have an application pending. It's before the FDA, but we have not received our 510K clearance and are unable to sell into the US market at this time. We do hope to have that soon, and we will certainly let our US customers know when that's available. So with that, I'm going to hand things back over to Jim, and he's going to give a brief overview of our current suite of lupus anticoagulant products and also talk a bit more about our HexLA test. Jim?
Thank you, Ian. That was a great overview. And it's really nice to be reminded of how long ago the hexagonal phase assay came to our attention back in the late 1980s. So thank you for bringing us back to where the historical perspective of all this testing is. So I'm now going to go into our CRYOcheck suite of lupus anticoagulant products. So here at Precision, as you can see, we offer quite an extensive portfolio of products for lupus anticoagulant testing. We have our lupus positive control, our weak lupus positive control, our lupus negative control. That's new to the marketplace. It is available here in US as well as Canada. We have our LA Check and LA Sure. Some of you on this call today are using that product, screening and confirmatory DRVVT reagents, the platelet lysate product, which is the source of phospholipids in the platelet neutralization procedure. I'll just drill into these a little bit more detailed information. So our lupus positive controls, again, we offer the CRYOcheck lupus positive controls as lupus positive, as well as a weak lupus positive control. They're intended for use as a positive or weak positive control in assays for lupus anticoagulant. The positive controls contain citrated human plasma. It's been collected from donors that have been tested positive in accordance with the revised criteria of the SSC subcommittee for the standardization of lupus anticoagulants. That's the ISTH group. It is available with a 3-year shelf life. It's stable for up to 8 hours. They both are stable for up to 8 hours after thawing when stored at 2 to 8 degrees C. And as with many of our calibrator and controls, we offer them in a 0.5 mil as well as a 1 mil format. So you've got more flexibility in how much of the product you need to use. New to the market, as I said earlier, is our lupus negative control. It's intended for use as a negative control in assays for lupus anticoagulant. It contains citrated human plasma collected from donors that have been tested in accordance with the revised criteria of the SSC subcommittee for standardization of lupus anticoagulants, same way as our positive controls. They are available with up to a 2-year shelf life. They are also stable for 8 hours after thawing when stored at 2 to 8 degrees C. And again, available in the 0.5 mil as well as the 1 mil formats. Next is our CRYOcheck LA Screen and LA Confirm reagents. So, excuse me, CRYOcheck LA Check and LA Sure are dilute Russell Viper venom time screen and confirmatory reagents. They're intended to detect the presence of lupus anticoagulant in citrated human plasma. These reagents contain Russell's Viper venom, phospholipids, as well as anti-heparin agents. They have a 48-hour stability once thawed if refrigerated at 2 to 8 degrees C. Unused reagent may be refrozen, and they come with a 3-year shelf life. So you've got good long expiry on that product. You can keep the same lot number for quite some period of time. Last up of the current offerings is our CRYOcheck platelet lysate. It is a source of phospholipids intended for use in the platelet neutralization procedure, which is used to detect lupus anticoagulants. The platelets are thoroughly washed to remove other cellular and protein contaminants, and they are then held at a concentration of somewhere between 250 to 300,000 platelets per microliter. They are then lysed by a freeze-thaw cycle. Last product in the mixture here is the CRYOcheck pooled normal plasma, which is comprised of platelet-poor plasma from 20 or more carefully screened male and female donors. They'll range in age from 18 to 26. It's recommended as a normal control for one-stage PT as well as the PTT assays, and it may be used as an alternative to your laboratory collected pools of normal plasma, such as what would typically be collected for use in mixing studies. So that completed what is our current panel of reagents and controls for lupus anticoagulant testing. And now I'm really excited to tell you about our CRYOcheck HexLA. That is our hexagonal phospholipid lupus anticoagulant test, and important to say to you, designed for automation. So I'd like to now get into that product with you. As Ian explained, we are pending FDA clearance right now, so we're not selling in the US. We are excited to have Health Canada have the product Health Canada authorized. So for you folks in Canada, we're very excited that we are able to present this product to you now. It is available for sale. CRYOcheck HexLA. So as a industry leader in providing coagulation diagnostic solutions, it was our goal here at Precision to present you with a validated hexagonal phospholipid anticoagulant test in a frozen format, and again, designed with automation in mind. So with this product, it now completes our full suite of lupus anticoagulant testing, from controls to DRVVT to HexLA. It is a complete package now for your laboratory testing in a frozen, ready, thaw and go format. So let's look at the HexLA and tell you a little bit more about it. There's no other hexagonal phase lupus anticoagulant test on the market that can match the complete offering of CRYOcheck HexLA. First and foremost, as with all CRYOcheck products, it comes in a convenient frozen format. It's ready to use within minutes. There's no reconstitution errors because there's no need to reconstitute any of the components in this kit. It is intended for use on automated coagulation analyzers. As I said, this product was designed from the ground up with automation in mind. And so what you're going to see is a real reduction in the waste of reagent that you'll have with this kit. What we've kept it in is a simple format, minimal number of vials, which means less preparation time so that you can get to results faster. We are unaffected by unfractionated heparin, as well as low molecular weight heparin, up to 2 IUs per ml. Some of the DOACs, dabigatran, rivaroxaban, as well as fondaparinux do not interfere with the interpretation of the CRYOcheck HexLA, nor does C-reactive protein. I wanted to point out to you a paper that we presented last year at the Antiphospholipid Antibodies Congress. It was in Manchester in September, where we compared the StayCloud LA to our HexLA, and we showed far better performance with high levels of C-reactive protein. I think that paper would be very interesting to all of you, and we're happy to provide it. As with all our products, it comes in a compact, color-coded packaging that makes for easy storage, as well as identification in your freezers. And we do have 8-hour stability on board the analyzer. That is based on the Star Evolution analyzer. And there is information on other analyzers as well. So let's have a look at the kit now. So starting out, the HexLA kit contains 2 vial sets of frozen reagents. So that gives you a total of 6 vials per kit. To keep in the theme of convenience, we are using color-coded caps. This is one set of reagents that we're looking at here of the CRYOcheck HexLA, and I'd like to go into each component with you now. We start out with our LA Start. That is a white capped vial. There's 1.5 mils of reagent in there. Now this contains the pooled normal plasma along with buffer and a heparin neutralizer. The next component is the LA Correct. LA Correct is a purple capped vial, 1.5 mils of reagent. It contains pooled normal plasma with buffer, a heparin neutralizer, and now the addition of the inverted hexagonal phase phospholipid. The third component, and final component, I might add, is the LA-APTT reagent. You'll notice this comes in a black capped vial. You'll also notice there's 3 mils of reagent in this vial. And Ian's going to talk a bit more about the fill of the APTT reagent and how much that accommodates automation. Now, this is a silica-based LA-sensitive APTT reagent with stabilizer. So I said there's only 3 components to our kit. If we take a moment to do a comparison of what a StayClot LA looks like versus a Hex LA looks like, you're going to see on the left side of your screen, StayClot LA, 5 components. Of those components, you see a format of liquid versus lyophilized. So you have a mixture of format here as compared to the right side where we look at Hex LA, where we only have 3 components: LA start, LA correct, and our LA APTT reagent. They're all in the same format: frozen, simply thaw and go. So by offering Hex LA in a convenient thaw and use format in volumes that are designed for automation, the amount of time spent addressing and preparing reagents is greatly reduced. And by incorporating the normal plasma with the heparin neutralizer in the start and correct reagent, we have further simplified and streamlined this reagent system. So if we want to look at the test format, if we want to compare how does it look to run a StayClot LA versus a Hex LA, if we go back to just looking at the actual tubes themselves, so on StayClot LA, you would add patient sample to both tubes 1 and 2. Now you have to add buffer to tube 1, phospholipids to tube 2, then normal pooled plasma to both, then your APTT reagent, add your calcium chloride, and start the reaction. If you come over to the right side of the screen, Hex LA has simplified those steps. You add patient sample. Tube 1 is now already containing pooled normal plasma with buffer. Tube 2 contains pooled normal plasma with phospholipid. APTT reagent to both. Add your calcium chloride. You're ready to go. So by consolidating the normal plasma into the buffer and phospholipid components, Hex LA offers the same benefits of an integrated assay with fewer components. And this really highlights the value of frozen thaw and go product versus lyophilized product. And that's why you see on the StayClot LA, you have to have the pooled normal plasma separate because it's lyophilized, it needs to be reconstituted. None of that goes on here. Much simplified assay. So now I'd like to turn this back over to Ian. He's going to talk to you a little bit about reagent preparation and the time that you will save and some of the stuff that we looked at back at Precision.
Thank you, Jim, and thanks for that great overview of Hex LA and our lupus anticoagulant products. So I just wanted to build on what Jim had said. We talk a lot about the time-saving and error reduction properties of our fresh frozen plasma. So what we did is we kind of set up a head-to-head as if you were going to prepare both StayClot and LA and Hex LA for use in a lab. This is certainly not a rigorous scientific study, but more of a quick look at what that might translate to as far as time savings goes in the lab. So you'll see StayClot LA on the top, Hex LA on the bottom. The first step for StayClot LA, you source your reagents, distilled water, pipettes, pipette tips, any other equipment that you might need, any other consumables. For Hex LA, you simply source the reagents and prepare your water bath. So no other materials are required. You might use one of our supplied foam floaties so that you don't get your hands wet. But other than that, you don't need anything else to begin preparing the product for use. For StayClot LA, the first step is you reconstitute reagent 2 with 0.5 ml of distilled water. And for Hex LA, you put the 3 vials into a 37-degree water bath for 5 minutes. If you're doing multiple vial sets, if your testing volume dictates, you simply add more vials to the water bath, where there's no change to that 5-minute time, versus StayClot LA, where if you add more vials, you need to address those vials individually, which does add time to the preparation process. So now for Stacklot LA, we're on to reagent 3, again reconstituting with 0.5 ml of distilled water. Hex LA, next step after the 5-minute water bath is to vortex all components for 5 seconds and allow the reagents to acclimate on board the analyzer for 10 minutes. So that vortex step is simply to make sure that everything stays in suspension and is nicely mixed on board the analyzer. So Stacklot LA, again, the next step would be to reconstitute reagent number 4 with 2 ml from reagent number 5, which is the solvent, and I think this is the APTT reagent. And then you need to wait 30 minutes and allow all of the reagents in Stacklot LA to acclimate for 30 minutes. And with one of the reagents, there's also a intermittent swirling step that must be followed during that 30-minute acclimation. Versus Hex LA, once the reagents have acclimated for 10 minutes, they are ready to use. So what does that look like overall for time savings? We worked out to about a 50-minute start to finish prep time for Stacklot LA versus a 20-minute prep time for Hex LA. Now 30 minutes is quite a bit of time over a day or an 8-hour shift in a lab. And you should also keep in mind that if you need to add additional testing, that's 50 minutes all over again. There's no shortcut to that preparation time, so that can add up if you need to go back and prepare more reagents. And also, there's no interface with equipment or other materials that could introduce error into the assay system. What's in the vial stays in the vial, is unchanged from the start to the finish. The only time you have to address the reagent is to put a stir bar into the APTT reagent. Speaking of stir bar, I just want to talk about that as well. Jim talked a lot about how this kit is designed for automation, and this is one of the features that we're talking about when we say that. Anyone who runs or spends any time using an automated coagulation analyzer knows that when you put a stir bar in a vial, you greatly increase the dead volume in that vial position. So when you look at the two kits, Stacklot LA versus Hex LA, we have gotten around that issue by simply providing more APTT reagent where the stir bar is placed than the other reagents. So it overcomes that dead volume, and you can thaw one vial, and on most analyzers, you'll immediately be able to get utilization without having to do any pooling or combining of kits. Now, if we look at the Stacklot LA kit, which has a 0.5 ml fill volume in the phospholipid reagent, typically what we've seen with our customers is that's not a sufficient volume to begin testing, and they are forced to pool either multiple vials or multiple kits to overcome that dead volume and then begin getting tests out of the reagent. So we've eliminated that need to pool. You still can pool if your volume dictates, but we've eliminated the need to pool multiple vials to get tests. And through that, we've greatly reduced the amount of wastage when you run this kit on an automated coagulation analyzer. So not quite designed for automation, but certainly designed for automation. And lastly, I want to talk about, before I hand this back over to Jim, is platform availability. I'm sure you're all wondering what platforms this is available on. And the answer is that we aim to have it available on all analyzers. We did most of our validation and design work on the Star Evolution, so a platform for that kit is available today. We're currently working on protocols for the BCS XP, ACL Top, and Sysmex CS series of analyzers. We do have this kit working on those platforms, but now is the task of validating the performance criteria and really making sure that those protocols are ready for use in your lab. So if you're looking for more information on timing of these protocols for the different platforms, please reach out to your AM, and they should be able to provide a bit more information on when you can expect those protocols to be available. So with that, I'll hand things back over to Jim. I think he has a few closing notes before we take questions. Jim?
Ian, thanks. That was a great way to see the value of our Hex LA kit. So appreciate that very much. And yes, Ian is correct. We just have a few more slides, a little bit more that we wanted to share with you as we close out the presentation. Just to speak about our educational webinars that we offer from Precision BioLogic. So we've still got some more webinars coming in the next weeks and months, and we wanted to share some of those topics with you. Our next presentation is titled Frozen versus Lyophilized, the Value of Precision BioLogic. That will be on June 3rd, 1 o'clock Eastern Time. Coming up in July is a really interesting and exciting area, the issues in chromogenic factor VIII testing, when we'll be able to introduce our CRYOcheck chromogenic factor VIII kit. We'll sort of round out the next couple of months with an update on factor VIII inhibitor testing, as well as updates on new CRYOcheck products. So we're really excited as we have many new products that we are working on. Now, if you're interested in being notified when these webinars are scheduled, you can reach out to us to make sure you're on our webinar mailing list. You can visit precisionbiologic.com/webinars. But again, if you're more comfortable, reach out to any of your sales representatives. There's always places where you can find more of the resources that we have available to you. Our website, precisionbiologic.com, is full of good information for you. There's a section titled Our Publications where posters are listed, all of the many posters we presented over the years at ISTH, ISLH, THESNA. I mentioned the poster from last September at the antiphospholipid antibody meeting. That's available there. Our webinars are recorded. They are available for you on our website. We pointing out here this one, which was the update on factor VIII inhibitor testing when we introduced our CRYOcheck factor VIII inhibitor kit. That was last May with Dr. Connie Miller, who was the former director of the CDC Hemostasis Laboratory. But as always, as Ian said, I'm saying reach out to your account manager, your area sales reps, and we're always happy to assist you in finding the information that you're looking for. Another valuable resource that is proudly supported by Precision BioLogic is the Fritzma Factor. It's your interactive hemostasis resource. And basically, if you have not visited it before, the website, www.fritzmafactor.com, it's free of charge. And George Fritzma, medical laboratory scientist, author, and educator, has started this blog back in 2007, and it's grown quite a bit, and it's an excellent place to exchange ideas and information about hemostasis testing. Now, we said always know you can reach out to your sales reps. We are looking for more ways to stay connected. Part of our job as sales reps is to get out there, visit with you, interact with you, share information. Our ability to do that has been limited now by this pandemic. So what we'd like to do is encourage you to connect with us virtually. Alyssa Rossman and myself, who cover the Western US and Canada, or Kelly Sweeney and CJ Crossan, who cover the Eastern portions of US and Canada. We're here to help you, to share information with you. So we'd be happy to set up a Microsoft Teams meeting or a GoToMeeting presentation to address any of your diagnostic needs. With that, we would like to open it up now for questions. Again, if you think of a question after we've ended the webinar, our email addresses are here, both myself and Ian's. And we thank you very much for joining us today. And now let's open it up for some questions. How does it look, Ian, for questions?
I'm having a look here, Jim. So I see a question. Somebody has asked, will the PowerPoint presentation be available? What we will be doing following the webinar is we'll upload a recording to our website, and we'll make that available to everybody. And we'll certainly send out a note when that is on the website. I suspect that will be available within the next day or so.
Okay, great. Thank you for asking that question. Ian, I've got one just come in here. Can you refreeze HexLA after thawing?
So no, you can't refreeze the reagents. It does have an 8-hour onboard stability, which is similar to Stakelot LA. Both reagents have an 8-hour stability claim.
Okay, all right. Ian, here's another one. These coming in pretty quick now. What is HexLA, when rather, excuse me, when is HexLA expected to be cleared by the FDA?
That is a great question, and I wish I knew the answer to that. So I can't say for sure when it will be available. What I can say is that we haven't just submitted the application to the FDA. It's been before the FDA for quite a while, and we are in communication with them. And of course, there's always the variability of what's going on right now with the pandemic. It hasn't seemed to impact the progress of our application yet, but we just couldn't say for sure when that's going to be available.
Yep, that's perfect, Ian. And I just would reiterate that data has been submitted to the FDA, so it's really that
Yes.
waiting game, right? When you
Yep.
rely on the government, I guess. Okay, Ian, I have another question. Do you have lupus patient comparison data for HexLA versus StateClot LA?
Yes, we do. So that was part of our FDA application was method comparison data for HexLA along with StateClot LA. We're not releasing that comparison data yet, just where we're still before the FDA, but I think we do have plans to release that as part of a poster or a paper at some point in the future. So that does exist, and we will share that with our customers as soon as we can.
Okay, well, that's great, Ian. I think that's all the questions we have time for. Again, if there's any questions we didn't answer, we're committed to get back to you via email. And if you think of any questions once we end this webinar, we're also happy, and we will make sure we answer all of your questions. So with that, we want to again say thank you very much for all of you to attend, and I'd like to thank our marketing team for putting on such a great presentation, and we really enjoy this chance to interact with you all today.
And thank you, Jim. It was a pleasure being on with you today.
Thank you, Ian. You take care, everyone. Goodbye.